Hi everyone, my name is Divya Narayanan and I'm here to present the topic Recent Advancements in Human Genomics. I'll be talking about clinical application of genome analysis. I am a trained diagnostic genetics professional and have worked in clinical labs in MD Anderson Cancer Center, Baylor College of Medicine, and Colorado Genetics Laboratory in past 3.5 years, conducting tests and analyzing data to reveal reportable mutations. I want to start by talking about the timeline and events that have happened so far in the field of genomics and how they ended up being clinically applicable. So the realization of existence of something changes making us better fit for our environment came from natural selection theory, following which Mendel showed us this something gets passed down from generation to generation. Then we came to know this something is nucleic acid. We also learned about chromosome structure and mitosis theory, and Morgan's fruit fly taught us gene linkage concept. It was Muller's experiment which revealed that X-rays mutate genes. So we also learned about transposons or mobile genes that travel from one position to other in within our genome. And Beadle tandem theory was also very good. It showed one gene produces one enzyme, although with time it has changed conceptually. Um, but breakthrough experiments were uh, Hershey Chase experiment revealing deoxyribonucleic acid as our genetic material, and Watson and Crick revealing DNA to be double helix structurally. Ingram's experiment brought attention to connecting the presence or absence of genes to a disease, and the disease in experiment was sickle cell anemia. This was the first stone in clinical genetics. All of these theories increased our realization, and the technological invention started with recombination, cloning, followed by sequencing. We started creating transgenic animals for experimentation and better understanding of genome. PCR paved way for speeding up our path to creating human genetic map using RFLP technique. With ESTs, that is expression sequence tags, we learned to tag the genes of interest. While we continue to reveal human genome map, we sequenced genome of lot of organisms that were of interest. And by finally in 2003, the human genome sequencing was completed and a revolution started demanding its application in clinic. 2005 was the year of GWAS, Genome Wide Association Studies, which started creating hope of banking genome data associated with phenotypic traits and diseases. We realized we have a lot of variations within our genome, which increase our susceptibility to a particular disease or decrease our susceptibility to a particular disease. There is not just one gene associated with a particular disease. There are many variants and many genes sometimes that can change our ability to recover or ability to get a treatment. So pharmacogenetic project was also done in 2005, which kind of described the drug response depending on genome variations in a particular human. So that year was pretty great. However, the interest in genomic diagnosis was not that much until 2010, I guess. Public eye and trust on genomic uh, diagnosis started after Nick Walker, whose boy with a very rare variant associated with a immunological defect, and this was identified by whole exome sequencing. And this boy got bone marrow transplant right after this diagnosis, which led him to escape life-threatening disease. So now things got really interesting, and also computational support for genomics started increasing multifold. The clinical demand got higher, but even if the sequencing was being done faster, the prices started reducing, technology started developing different ways of sequencing and low cost sequencing products. The bioinformatic infrastructure started lagging, and that was due to lack of trained professionals and also lack of databases. So servers and databases started flourishing. People started storing their the growing sequenced genomes, and publicly sharing them. And by 2014, we have for $1,000 a genome can be sequenced. That was the level we reached. The cost of bioinformatics still is a hurdle, but I'm sure with more and more advanced analytical pipelines, the cost is going to decline. But large scale clinical sequencing projects, which are also extending to pharmacogenomics for companies like AstraZeneca, which launched a 2 million genomes project for their own clinical trials. They aim to incorporate these genomics into clinical trials and revolutionize pharmacogenomics. And with many other additional companies and studies uh, who also follow their footsteps. It's gonna be great year ahead and great time ahead, but generation of genomic data also comes with a lot of ethical and legal questions. And unlike other medical interventions, genomic data has a direct implication for individuals, and not only individuals, but the populations which are beyond this individual who is getting tested. So you can reveal genomic changes which are particular to a particular population, and changes the dynamics and relationships among everybody. Recently, in 2018, NIH has invested heavily in sequencing, and it has moving projects like TopMed, which is transomic for precision medicine, and they are going to create database for more than 100,000 whole genome sequence individuals with their detailed phenotype and their clinical records. This would be a great step for clinical genomics, because the most of the biggest challenges are to connect the phenotype with the genotype, because we have the genomic sequencing undergoing massive expansion and having a lot of data stored. It's a rarely approached tool in a clinical setting yet, because of the lack of databases and information. So next generation technique is basically massively parallel reactions and arrays. What we were doing in a tube with one probe, one sample, um, we are now able to do in a chip with millions of probes for one sample, and we can do more than one sample in one chip. So the concept started with microarrays, wherein we have immobilized probe, we add the DNA, we have gene of interest as probe. So we check if the particular region is present in the patient DNA or not. Um, but then we revolutionized, and there was sequencing arrays coming up, which kind of synthesized the DNA right there on the glass slide, um, to see what exactly the sequence of the patient DNA is, so that we know any mutations, change in variants, and everything. Now, the latest concept is mapping. If you are aware of cytogenetics, I mean, karyotyping has been running from probably the years the chromosome was discovered. Uh, and Even today, cytogenetics is the norm. It has to happen in oncology, especially. Karyotyping is the first step to see if there is any structural variations. Uh, it's the norm test that everybody does. So mapping is challenging that karyotyping after ages and ages and ages. Pretty much people in the field of cytogenetics have been saying that we don't see karyotyping dying in the future years. And next generation mapping is something that is brought by BioNano company, and they believe and they claim that they can provide us with the proper breakpoints and everything. You don't have to sequence everything, and see it's cost effective, and it is very meticulous in the breakpoint. So it covers insertions, inversions, deletions, duplications. So it's frame shift mutations, everything, and it's interesting that they claim to do that. They are still applying this technology as a research basis because there's a lot of the bioinformatic efforts. So the company is still struggling with the bioinformatics, and that's why the clinical applicability is going to take a few years. But looking at the era right now, I don't think it would be too long a wait to replace karyotype. So if this technology um, does what it claims, I'm sure a new set of bioinformatic tools are gonna be in the market soon. So there is a lot going on just with next generation. We have a lot of uh, sequencing techniques out there, right? We have target sequencing, we have whole exome sequencing, whole genome sequencing, RNA sequencing, immunotherapies. We have microRNA, long non-coding RNA sequencing, cell-free DNA sequencing. But clinically, uh, we are mostly using only target sequencing because we know what we are looking for and it's easy for us to retrieve bioinformatically. Whole exome sequencing is slowly getting into the clinical applicability with some, filters, which is unique in each lab. RNA sequencing is only used when DNA sequencing is not giving an answer, or we want to follow it up. But it's harder to do RNA sequencing clinically. Um, we require a lot of training, a lot of bioinformatic help and understanding, which is not there yet. So clinical usage of these sequencing for diagnosis is pretty less. So the clinical genomic sequencing, what exactly the workflow is, right? We get a patient who has some symptoms, or in case of oncology, it's a slow progressing disease. So sometimes people only realize it very late. So the patient comes to the clinic for diagnostic purposes. Sometimes it is for family history because somebody has a genetic disease in their family, or sometimes it's for screening for pharmacogenomic purposes. But most of the times it's diagnostic because people are less aware about the other stuff. Once the, the patient is in the clinic collects these information, medical history, family history, physical examination is done. They are given genetic counseling. They are also, you know, uh, taking insurance into the consideration because insurance has to cover the expensive sequencing technique and pay our checks basically. But this is a US system. And not every clinic does the genetic counseling. They sometimes outsource it. But the family history and medical history is absolutely necessary in medical practice. So they do collect that information. Now, how much they share it with us is up to them nowadays.
So that makes our work really difficult because a medical practitioner would see so many patients in a day. He would not spend time writing all the symptoms. He will just order the sequencing and he will write two or three stuff. Okay, the patient has this, that, that. But he will not describe the entire phenotype of the patient. So we will get 2 to 3 words of clinical indication information. Now in the lab, there is sequencing conducted. Sequencing is not the only test that is conducted in the lab. When a sample comes to an oncology testing lab, clinical lab, they will have other tests also for the same sample. But let's concentrate on sequencing, right? So sequencing is done by a person. He will conduct the entire technique. He will do the sample processing, everything, and the results raw data is coming. And also we will have the raw data filtered. We'll do the sequence alignment, variant calling, variant filtering, everything. Now after that, the same technologist is gonna sit and give a brief outline of gene and phenotype association, or he will conduct a basic variant analysis, which is like he'll have a set number of the databases. He will just go through all those databases, check what makes sense for this patient, and just write that down. It's then the senior people or research oriented people or supervisors. Basically, my work is to go into deep detail. So we conduct even more deeper searches of database. We will conduct deeper searches of PubMed and research papers and contacting other clinical labs that we know if we have some rare variants of worry and everything. And we create the major report that sometimes in my lab I had a genetic counselor, but it's not necessary that every lab has. So that will directly go to the clinical laboratory director who would try to make sense of all the other tests and with this sequencing results and try to make combine suggestions for the clinician and for treatment and send the genome report out. Now, once the genome report is out, the clinician is going to sit and make a medical decision. He will try to rephenotype. He will see if he can tell us something else also and let us, know to reanalyze. So many a times the thing happens that he will talk to the patient all over again, and he will ask about the family history. He will tell about this and that. This is what we see. Do you think somebody else has it? Do you think you saw these symptoms in you? Or he will talk even more detail, and then he'll come back to the lab and he will ask us to again re analyze the sequence and find certain medical questions that would uh, you know kind of pave his path to treating the patient. So we will redo it, and this time we will try to use a variety of other databases or PubMed sources. All in all, the goal is to provide a modified treatment, a personalized disease surveillance, and uh, that takes around 2 to 3 weeks for a patient, for a particular patient, in real time. In reality, that is the time that you are looking at. So, exactly what happens in a typical genetic testing lab, at least in Colorado Genetics Laboratory, it was we had cytogenetics and molecular genetics in the same floor. So this is our workflow basically. When a clinician orders an oncology related testing, uh, he is going to order the karyotyping, fish for mini probes, 1 to many probes, and either a SNP microarray or a del test. If he's very focused on what he wants to know, is he's pretty clear on what the goal of the diagnosis is. And if something comes to know in these two, and he wants to sequence that region, or he does not get an answer, and then also he would still go for the sequencing, and then he would order for a sequencing. First, he would order for a targeted, and then whole exome sequencing, and then if that also doesn't give him any answer, he will order for whole genome sequencing. But the first few tests, if he has some idea of this is the genetic aberration that is happening, we get some answer, he will start the treatment with what he knows. The sequencing kind of gives him, um, another feather to his cap. But sometimes they will not do much. They will just order for karyotyping fish and then directly to targeted panel, right? Because that's the easiest. They have well experienced staff, so they know exactly what is going wrong with the patient, so they will order for these things, which is great. Once that happens and we have something that we want to answer, we want to confirm it also. So we mostly, a lot of the tests, once they undergo sequencing or microarray or something, they have another confirmatory test that to be done, Sanger sequencing or fish or real time PCR. So once that is done, we will still continue to follow this patient. So patient sample keeps coming to us while they are in treatment, after they are in remission, in if to their luck, if they have a relapse again, the same patient will come to the lab for the same kind of testing. So we have a lot of data on one patient from the time their first sample comes to the time they get treated or have a relapse. And every oncology related case comes back to us. It should come back to us. That's how their genomic profile will be kept intact, and in realistic times it's easier to treat them. The people who are in a particular lab involved in case flow is technologist who conducts the technique, who will conduct the experiment, who will collect the raw data, do the variant calling and filtering and everything, and the first step of variant prioritization, and then it would be us to do the deep research analysis of these variant, and we will have back and forth discussions with the genetic counselor as to what is possible for them, what are they talking about when they say these medical terms, what does it mean in this database, this is the term that I see, it's not the same, but does that mean the same? So these kind of education we get from genetic counselor. Very few labs have genetic counselor. Most of the time, it's people like us who are trapped in both sides, on the technical side, on the medical side, who have to converge all of this knowledge and put it together, and then do the research and find the variants and make sense out of it, and then report it. um, it's time consuming, and our brain cells get saturated at after a point, because we need a lot of bioinformatics support, and we are right now not at that point where bioinformaticians would prefer to come into a clinical lab and work, because they don't know the drill, and the pipelines are not that well established as to what the requirement is. Each patient has a different requirement. Each patient has to be individually looked upon for each and every variant. So there is so much of permutation combination that it becomes very difficult for a bioinformatician to do his job, concentrating on he would rather prepare a genetic pipeline, and every time he will have to troubleshoot it. So we are not there yet, but I am pretty sure that there is a lot of training programs going on, a lot of people coming up. So it's gonna be a brighter tomorrow, and somewhere we will have bioinformaticians in the clinical. And clinical laboratory directors' work is basically to bring all of these tests, not just sequencing, everything together, and put a medical sense to it. So there are three types of sequencing that we can do, right? Targeted panels, whole exome, whole genome. Targeted panels are the most clinically easy to apply because they focus on certain number of genes. We know what we are looking for. We know whether we have to just, it's a yes or a no answer for each gene, right? Mutated, not mutated, what kind of mutation, and how does that affect. Only 3 4 questions per gene. We do have panels up to 200 genes. Not everybody has everything, but it still makes whatever test they have, it makes it easier for us to do the bioinformatic part and get some valuable answer for the clinician. But it's easy. But it does not cover everything. So it does not. If we see something, we don't know if something else is also going on wrong with this patient, and going to affect the drug response, or going to affect the gene's action, or on something or those lines, it affects the disease progression or not. And if we fail to have a targeted panel, for none of the mutations work for certain cancers, so then we have to conduct a whole genome or a whole exome. Whole exome is getting more reputation in clinical labs nowadays, because now we can, it's in the middle, so it can, uh, we have a targeted list of genes that we look for, and if we don't have any answer from that list, then we can expand, right? We don't have to conduct another test for the patient. So it is research as well as a little bit of clinical also, right? It's feasible, but it has a high bioinformatic effort. We haven't reached a point where we can easily undergo a database screening, PubMed screening very easily. So it's very time consuming when we have to do a clinical interpretation. There are unknown variants that come up, and it becomes really taxing and on our end to read through. So if whole exome is so difficult for us to have a answer for the clinician within a time frame, whole genome is out of the question. So whole genome is the last thing a person would order if if none of these give him any answer. And rarest of the rare cases go for whole exome, whole genome. Mostly it is because it is expensive than other tests, and also it does not have any certain value. So for clinical interpretation, it's we have long list of unknown variants in this, so it becomes really difficult for us to concentrate on what is important, to filter out what is important. The curation is really difficult.
Search. Uh, these are few databases that I've written down. There are plenty of databases. There are tons of blood databases that you can go into. These are some very, very promising databases, which makes sense for a clinical lab to go through and based on their requirement, though. So, Civic is a new database. It's a clinical interpretation of variants in cancer. It kind of is a public domain. Anybody can put in their findings, and the Civic group kind of also gives us the. They will analyze the contribution, and they will tell whether the evidence is trustable enough, and whether it is clinically applicable. What is the level of evidence? So there's 5 levels of evidence. Fifth being the most strongest evidence, to zero being, it's probably not very reliable. Um, then we have PMKD, Precision Medicine Knowledge Base. It's a very straightforward database. There are only few genes which definitely have some drug treatment, and checking that out also helps in a clinical lab to add information for the patient. There is a database of curated mutations. Again, this curated mutations, they just follow what is the mutation. So there is a variant, and the contributions talk about a little bit about their clinical implication, and sometimes also about the drug treatment. But mostly it is about mutation. So it's also a research, I would say, research database. Cosmic is pretty old, and Cosmic has been the most commonly used. The reason being, Cosmic gets updated every 6 months. So every 6 months, they would conduct an entire research of all the papers, new mutations, and everything, and they will add it to the Cosmic. So it's the most updated database. So that's why people love to check catalog of somatic mutations in cancer, and it also differentiates based on where the sample has come from, whether it's spleen, whether it's liver, whether it's coming from the colon, where the tissue is coming from, what kind of cancer is it. Um, it has a direct discussion of the path. It has the direct discussion of whether the mutation is a nonsense mutation, it is a missense mutation, and then it also talks about its association with patients cohorts. So it's good, uh, but it has you need to know the tactic of finding your questions answered. We have PQM, which is pediatric cancer genome. It's also pretty latest and new, um, and it is well followed nowadays slowly by the labs who conduct childhood cancer testing. Um, International Cancer Genome Consortium is a effort towards having all of these databases combined, and worldwide, whoever is conducting the test, whoever has found finding, whoever has discovered a new thing in this field can contribute to this database. So it's a very good field for collaborations, and also gives a lot. It has a comprehension of many other databases, so it's a new effort as well. Jack's CKB is also pretty old. It's a little old. I would not say very old. I mean, these are all after 2015, 2010, and all that. The Jackson Lab has a clinical knowledge base, and they have really great research done in their lab. So um, they do a lot of clinical interpretation for whatever they do in their lab. So it's pretty interesting, and it has good knowledge base for us to follow. Uh, there is C Bioportal, which was started by MSK in New York, and they have C Bioportal has again a lot of information. It is a little older database, so it has much more collection. It is also given by MSK, so their experiences, the patients who come to MSK are varied diversities, and even the rarest case land up in the MSK and MD Anderson. So it helps. There's a biological connection that they also provide us with information. So that database is pretty good for us to clinically use. OncoKB is precision oncology knowledge base again. Um, you know, just like PMKB talks about the drugs that can be applied for certain genes, which are oncogenes. So it's a helpful database. Uh, Integrative oncogenomics is pretty new. It has all various mutations, and from structural, also somatic, germline, deletions, duplications, indels, everything comes in this. and it kind of gives a chart of genes role. CGI is a fourth from Cancer Genome Consortium, and it's a cancer genome interpreter. So it's very new, and the goal is to have to help a clinician, laboratory director, and people in the lab to just enter the mutations and the details of the phenotype, and then kind of give us back, interpret the results easily. CCGA is again one more database, but this is more on the cytogenetics part. It has a lot of genomic aberrations, uh, gives us. Also, middleman is also
So, precision medicine, basically getting a treatment, based on the genome profile for a patient. It requires a lot of informatics infrastructure, not only software but also hardware level infrastructure. This is the ideal situation that we require. I mean, we require data storage, proper data storage, IT support, bioinformatic pipelines, and we want all of these people who are dealing with these data to be trained on how to use it, how to troubleshoot, how to get what they want, uh, and have accessibility also between them. Um, but that's not, I mean, this perfect scenario is not right now, which is happening. From the big data, I mean, for large volume of patient data, especially in oncology, the large volume of patients data is disseminated across a large variety of databases, and in order for us to extract this data, we face a lot of challenges. We have technical level challenges because we don't have powerful computational architecture, both software and hardware. The organizational and management level for defining these procedures on how to collect the data is not of good confidence. It's not of good quality, or we cannot easily trace them. The scientific level also is very very difficult. I mean, if there is we need to have more sophisticated mathematical models, so that we can predict the evolution of the disease, we can predict the risks for the patient. So definitely an efficient informatics architecture in supporting PM is required at this point. Um, and therefore I would definitely say that for the future of clinical genomics, we need the bioinformatics. It's the most important bottleneck if we have to get above these challenges and have the routine application for precision medicine at ease.